Good afternoon to everyone. I'm Valerio from Charles University in Prague, Czech Republic. And today I'm going to introduce to my project test of genetic code evolution hypothesis, so reverse evolution of the RNA binding domain of the ribosomal protein L11. Okay, first of all, I introduce like how was evolved with, hypothetically was evolved with the genetic code. Different theory assumes that the original genetic code coding only for a small set of amino acids. And this small set of amino acids were introduced like in the ancient prebiotic protein. But how the scientists like decide which amino acids are introduced, are like early one and which other one could be like late amino acid? Particularly this guy, Higgs, Podris, like summarize all the different theories and also all the different evidence about the abundance of the different amino acid in, for instance, in the Miller experiment, meteorite, or in the presence of hydrothermal vents. So it just classify all the amino acid in basis of their abundance in this environment. And here in the table we can see that we can split the chart into two parts. The first part are represented like the early amino acid. Instead, the bottom part of the chart is like the late amino acid. We can also find like some evidence directly in the genetic code. In fact, we see that in green is represented like the early amino acid represent more than 60% of the coding, the codon that coding for amino acid respect to the late amino acid. But for the moment, are just like just theory. So no one has demonstrated experimentally these different theories. So my project is to verify that it's possible that a protein composed of only early amino acid is still maintain a function or a structure. So how to do this? Here more or less is the flowchart of my project. So the first step is like the selection of a target protein, like a modern protein, with some function could be like an enzyme or like a binding protein. After that, generate a library composed only of early amino acid. So substitute only the late amino acid with the early one. And the next step will be the characterization of the mutant. So understand if, for instance, the mutant maintain the structure, maintain the function, or both, or nothing. Okay. So the first step was the selection of the target. To do this, we have to follow some strict criteria. The first one, the protein, the target should be small. So between 50 to 100 amino acids. This is more related to the more practical part, because of course, if we have like a small protein, more than 100 amino acids, we will generate a library of like more than 10 to the power of 30, 40 mutants that it's impossible to use like in practical way to manage this kind of library. Second step, the protein should be like conserved. And also this step, this criteria is really important in order to reduce the size of library, because with like, we can use like multiple sequence alignment or like a structural alignment in order to reduce the size of the library. Third step, criteria is that this target should be maintained like should have like the early amino acid in the most critical position for the function or for the structure. This is quite obvious because, for instance, if we selected, I don't know, like an enzyme with arginine in the, one arginine or lysine in the active site, and we substitute the arginine and lysis, it's really probable that the protein will lose the activity. So we need to find also someone that maintains the early amino acid in the critical position. After that, we need a target that is well characterized and with the possibility to have like a selection method, so to select the mutant, so to perform like practically. And the best target it was chosen was like the RNA binding domain of the ribosomal protein L11. This domain is composed of a small domain, 76 amino acids, so correspond to the first criteria. Is like its function is to bind a specific sequence of RNA from the ribosomal RNA. It's highly conserved, of course, it's a protein of the ribosome, so it's strictly conserved in most of the species and in particular the amino acids that are going involved in the interaction with the DNA are mostly early amino acids, except for instance, lysine and arginine, but most of them are like glycine, serine, so it's really good candidate for the test. But also, if it's small protein, still the library will be huge. Like we can talk like 10 to the power of 30 variant if we won't substitute all the late amino acid with the early one. So in order to reduce this one, we have performed like different multiple sequence alignment or also structural analysis. For instance, we can reduce the size of library. For instance, in RBD, in our target protein, for instance, in some spot could be like an arginine, but we can find that in a really homology protein, there could be like an aspartic acid, and these mutations still maintain the function. So we can reduce the size of library in this way, just with a multiple sequence alignment, or for instance, depend on the position of the late amino acid. If, for instance, if a late amino acid present is like an alpha helix, we can, for instance, avoid like the presence of a proline or other kind of amino acid. At the end of this analysis, we have reached like the size of 10 to the power of 10. That is still huge number, but with the mRNA display technique, it's still possible to express this kind of library. This technique is really powerful because respect to the other one, for instance, like the phage display or the technique that you can express a library maximum of 10 to the power of 9 clone. In these cases, we can express 10 to the power, so 3 magnitude more respect of the other technique. This technique uses it practically connect together the genotype to the phenotype. So we have like the RNA, the protein, linked to its mRNA. How it's possible? Thanks to the puromycin. And these molecules, so during the translation, the ribosome, when arrived to the 3-terminal, interact with the puromycin that enter inside the ribosome and link the protein. And in the same time, disassembled the ribosome. So we have like the RNA linked to the protein. But before to perform the mRNA display technique, we have to optimize the selection method for the protein, and we have optimized with the wild type. So we use it like a cell-free expression system. So it's meaning that we didn't use the cell, but just the extract of ribosome and all the translation equipment system for the expression. So we will express the protein was expressed, and after that we're incubated with the target RNA that was functionalized with the biotin molecules. After that, for the selection, we can was like the complex protein and target. So we're incubated with the streptonin bits, so it's possible to select the complex. And here we can see like the Western blot, like gel, where like in the first lane we can see like the flow through. So all the protein that doesn't bind the tRNA, and it's empty. So it's meaning that all protein interact with the RNA, with the target RNA. In the washing step, so it's nothing. So it's meaning that no weaker binder, so all the protein binded. And in elution, we can see our product. It means that this protein, this selection method is, at least for the wild type, is still available. So we have introduced this selection method inside the mRNA display technique. Here is like how to explain just like the flow chart of this technique. It's basic. Start with the DNA. So we have like the DNA library. After that, we transcribe the DNA library in RNA. After that, there will be the step where the RNA will be linked to the puromycin molecules. And in the linker, there will be like a fluorescence probe that will be useful for the next step, just to follow like the different step and the presence or not of the protein. After that we have this construct like RNA plus puromycin. There will be the expression, so the junction between like the protein and the RNA. And after that we have like this library composed of like 10 to the power to the 10 to the power 10 variant that will be selected toward the target RNA. After that, there will be like the elution and the step of reverse transcription. Because here we have the RNA. In this step, we need the DNA for the sequencing or a new round or to perform a new round of selection. So there will be like just reverse transcription, so from the RNA, DNA. It's important also the negative control, because I don't know if someone worked with the RNA, but the RNA is really sticky. So we have to subtract the noise from the project. So more or less for the negative control is the same step, is the same flowchart, the same parameters. The only difference that the library will be incubated not with the target RNA, but just with the bits. So in this way, we can select the protein that bind unspecifically the surface or the streptavidin or like the complex. And after that is the same process. Okay, here we can see step by step, because the mRNA display respect to other technique is not something like a commercial key. We need to perform and to optimize every single step. Here is the first step, so the ligation between the RNA and the puromycin. The puromycin contains a fluorescence molecule, so we can visualize in fluorescence, and we can see like the free purotag, so, and in the top of the part, like the conjugated mRNA to the puromycin. On the other side, we can see the visualization with like the common UV visualization with the gel red, and we can see that the yield, it's more than 80%. It's meaning that still some RNA does bind the puromycin, but the process is still okay. After that, the RNA will be expressed, will be translated, and here we can see the difference between before and after. So only mRNA, and after the translation. So we can see a shift, that meaning that there is like protein plus mRNA. So there is like expression. And after that, the selection. So here is represented like the flow-through. So all the library, all the variants, mutants that are not able to bind the target RNA, so lost the function. And it's the most abundant, of course, because no folding or like protein are not able to bind. The washing step, so just to remove like the weaker binder, and dilution. So just this one, it's meaning that some protein maintain the function. So composite of all early amino acids still maintain the function to interact with the target RNA. After that, there will be the reverse transcription. After the reverse transcription, this is like the visualization of the reverse transcription. So after the elution, will be retrotranscribed, and we have compared like the negative control, the sample from the negative control, so without tRNA target, and the library. We can see at the beginning that round by round, because it was performed like 60 times, there was like a noise. But after several times, we start to decrease sensible the noise, but still there. So it's meaning that there is like a subpopulation of protein that does not bind the target RNA, but only the bits. But to understand which was like this kind of sequence, we have sequencing, the library, and the negative control. And here the result of the sequencing from the next gen sequencing. And okay, of course, the best binder was the wild type. I don't know how it was possible, but there was like a contamination from the first round. And so the best binder, of course, is the wild type. But at least it's meaning that the method, it's robust. I mean, because the purpose of our method is to select the best binder. And the wild type, of course, is the best binder. But here instead, we can find the contamination, the noise. We can see that round by round, this sequence is equal present abundance in the same abundance in the negative control and in the library. So it means that stick to the bits. But we have found other three sequences, especially the sequence number 9, that has a really, really good ratio between library, so the samples with the RNA, and the control, negative control. That means that this sequence, this protein, does not have the tendencies to bind the negative to the bits. So it's really probable that this protein is still maintain the function. So bind the target RNA. Here we have like there was like the alignment of the different the selected protein respect to the wild type, and we can find that some mutation were common in the different clones. So, for instance, in some cases, really interesting. For instance, like there was like a substitution. For instance, in the late 40s, like the arginine were substituted by aspartic acid. So there was completely a change of charge in our binder. Furthermore, there were during the reverse transcription, there were also were inserted other mutations. We can see that, for instance, in the red arrow, there should be like were substituted like early amino acid with other early amino acid. This is a spontaneous process during the reverse transcription. But this mutation were still maintained and still maintained. At least, yes, like this is previous just homology modeling, so we can see that at least at the first analysis, it seems that the mutant maintained the function. It seems that maintained also the structure. But for the moment, this previous data is just homology modeling. So, okay, in conclusion, the future work will be like, of course, the expression of the single protein, characterization just to understand the binding constant with the target RNA, and of course, the crystal structure with the RNA. So in conclusion, we have identified a robust target for the reverse evolution project. Was customized the mRNA display technique for the best mutant selection, and it was identified 3 different proteins able to bind the RNA also in presence of only early amino acid. I'd like to thank my group from Charles University, and in particular Professor Kosuke Fukushima from the ELSI in Japan for the collaboration, and thanks to you.
Questions for Valerio?
So you identified as a candidate this protein because it had a bunch of the early amino acids at important positions. Are there classes of protein that have a larger tendency to have important sites, be these early ones versus later ones?
No, they are proteins that contain only early amino acid, and we can cluster in some pattern, but all the sequence that we have selected contain only early amino acids.
No,
so
what I mean is when you're looking at existent proteins, I mean so you're saying there are existing proteins that you can grab today that only have early amino acids.
In the prebiotic world, it was plausible that it was some protein like that.
Right,
but no evidence. I mean, it was just basing on the sampling on the meteorite and on the prebiotic world, but no protein was found that contained only early amino acids.
But I'm wondering if there are modern protein classes that are more biased towards early amino acids than later today. Maybe if there are functions in the cell that seem to be biased that way.
For the moment, no, it's not much.
Other questions? Ooh.
Thank you for your presentation. I realized that you already have a lot of possible combinations in your essay when you start, but I was wondering if you ever considered adding prebiotically relevant amino acids that we do not find in proteins nowadays. For example, ornithine could sort of solve the problem that right now you're not able to use any positively charged amino acids.
Yeah, this is the first step. So we have selected only the early amino acid present in the genetic code. But of course, the most theory provide evidence that was composed the early amino acid of conventional, like the modern one, so the present is, but also non-conventional one. For instance, one of the cases like the ornithine, in fact, if we notice the table was not positive charge amino acid. It's quite weird. So it's possible that the ornithine were replaced after that one. So, but that will be the next step of the project. So first step like this one, and after that we see to looking for the non-conventional. But this is quite difficult because you need to integrate in the translation system the possibility to insert non-conventional amino acids. So it's not so easy to modify the tRNA synthetase and blah blah blah.
Here, thank you very much for your talk. I assume that the positive charges in the proteins are interacting with the negative charge of the phosphate in the RNA. But in your new proteins, several of those positive charges, arginines and lysines, are now negative, like glutamine and aspartic acid. Do you have any idea or any model of how now these negative amino acids are interacting with the
Okay.
RNA? Thank you.
the only part that interact with the DNA is particularly this alpha helix and this loop. And mostly, okay. And this alpha helix and this loop is covered in this part. So this is remaining practically the same. And probably this, what is like, for this positive charge was on the other part of the alpha helix, so it's not interacted directly with the tRNA. So probably it's not important for that binding. And also, it's also possible that, I don't know, maybe some positive charge like the cation can function like a bridge. So it's also possible. But I know this is only homology modeling. But next step will be to have the crystal with the target RNA. Me too, I'm curious to know how it's like fit. So future work, I hope that will be soon.
Okay, let's thank Valerio again.